Thank you so much for listening to my thesis defense today. I'm sorry that we cannot all meet in person. This is the best that we could do. So I'm going to go ahead and just get started. So my research is entitled The Characterization of CDC42 and the Presence of the Small Molecule ATA197. My name is Meredith Ott, and I've done all this under the direction of my thesis mentor, Dr. Paul Adams, in the Department of Biochemistry. So just a little bit of background information. Our lab works with a superfamily of proteins that are called the RAS family of proteins. And so that RAS superfamily is divided into a bunch of different subfamilies. And so we work in the RAS superfamily and in the Rho subfamily of proteins. And so what these are is that the RAS family is a group of 150 small GTPases that play a huge role in a variety of cellular processes, such as cell growth, apoptosis, and signal transduction. So this figure shows some of the Rho proteins, just some of the 150 proteins that the RAS superfamily covers. So research normally refers to these as molecular switches, and this is because of their ability to switch between an inactive guanine diphosphate bound state and an active guanine triphosphate bound state. And we'll talk about the activation a little bit later. But our lab specifically works with the protein CDC42, or cell division cycle protein 42. It's in that RAS superfamily and in the Rho subfamily. It's 21 kilodaltons, and it acts as a molecular switch in a variety of signal transduction pathways. So what that means is that CDC42 is able to go on and activate other effector molecules, and then these molecules can have an effect in the cell. And so it's of importance to us because CDC42, the abnormal expression of it, has been found in a variety of cancers. So this is some of the effectors that CDC42 can act on and their normal cellular function. So the effectors of CDC42 play a wide variety of functions in the cell, such as cell-to-cell adhesion, cell growth, cell apoptosis, all of these things. And so it can seem a little bit obvious that if CDC42 is overactivated and all of these things are overactivated, that can lead to some negative consequences in the cell. And it does. RAS proteins in general have just been found to be involved in cancer. 30% of cancer cell types have some sort of RAS abnormality. CDC42 specifically, its underexpression has been found in adrenal, kidney, and pancreatic cancers, and it has been overexpressed in cervix, lung, and thyroid cancers. And so that is because of the activation. You can see from this diagram that when CDC42 is activated, it can go and activate all of these other pathways, and those can lead to some oncogenic effects, such as tumorigenesis, tumor metastasis, cancer cell migration, all of these negative consequences. So obviously we're concerned about how CDC42 is activated. And so it's activated through this GTP and this GDP cycling. So there are a couple of different proteins of interest that we look at when we talk about the activation of CDC42. First are GIFs, or guanine nucleotide exchange factors. What these do is that they exchange a GDP on an inactive CDC42, they exchange it for a GTP that's activating CDC42, and then it can go and activate other effectors and lead to a cellular effect. CDC42 is inactivated by GAPs, or GTPase activating proteins. They hydrolyze the GTP to GDP and thus inactivate CDC42. The last protein that we look at are guanosine dissociation inhibitors. These take an inactive CDC42 and lock it in that inactive state. Thus, it's unable to exchange for that GTP, it's unable to be activated. So there are a couple regions of the cell that we are of CDC42 that we are focused on in our lab. The first is the switch 1 and 2 regions. So these are two important regions of CDC42 that exhibit conformational freedom. They're able to change shape and thus bind these effector molecules. So there's a switch 1 and a switch 2. Switch 1 is the primary binding surface. So it has the most conformational freedom. It has the most affinity for all of those downstream effectors. Switch 2 is important in binding. It helps bind, but it is thought to be more important in binding the gamma phosphate and stabilizing GTP whenever it is bound. So these are two important regions. We also look at a mutant of CDC42 entitled T35A. So the 35th position in Ras proteins is very important. It's invariant throughout all Ras proteins. And this is because of its importance of binding GTP. So this mutation takes at the 35 position what is normally a threonine, and it substitutes it for an alanine. So it goes from this hydrophilic to hydrophobic molecule, this mutation is located in the switch 1 region. Studies have shown that the T35A has less conformational freedom, it has less binding ability for those downstream effectors. So we're really interested in seeing differences between CDC42 and the mutant T35A. We're also interested in this small molecule inhibitor of CDC42 entitled AZA197. So this molecule has been known to inhibit the activation of CDC42, and it has been known to inhibit cancer growth. So in a study with mice who were injected with colorectal cancer cells, the mice that were subject to this AZA197 had better prognosis, increased survival, and a shrinkage of tumors compared to the control group. So we are trying to understand better how AZA197 can affect CDC42 and play a role in its signaling. So in order to do all this, we first have to grow and express our protein. And so how we do this is that we make a C culture of E. coli cells in a LB broth media. And these E. coli cells contain a plasmid that have an ampicillin resistance gene and a 6-amino his tag. We'll talk about that in a little bit. So we let that sit overnight, and then the next morning we inoculate our media with our C culture of cells. And so then we let it grow for a little bit, and we let it grow until it is at an optical density of 0.6. And so that is when we know that it is in the log phase of growth. And so once we know that it's in the log phase of growth, we induce transcription with IPTG, and this allows us to overexpress our protein in these cells. So once we do that, we express the protein, we collect it, and so then we want to purify it. We want to make sure that we have no contaminants. So what we do is we use a fast protein liquid chromatography. And so this is a protein affinity chromatography, and how it works is that we subject our protein to a column. And so first we allow our protein to bind throughout the column. And how this works is that the column has these little beads that contain nickel. And so that 6-amino histidine tag that I talked about has a high affinity for nickel. So it binds to the nickel in the column, and it allows it to interact. And so then we wash everything else that is not our protein through the column while our protein of interest is interacting with those beads. And then lastly, to elute, to get our protein out of that column, what we do is we use an elution buffer that has a really high concentration of imidazole. So how this works is imidazole also has a high affinity for nickel, and since we are subjecting our column to so much imidazole, it binds instead of our protein, and our protein is able to be released from the column. So this is an example of one of the FPLC readings that we got. So what happens is that it measures the UV absorbance as far as intensity. And so the UV absorbance goes up because of our buffers, and then it goes down. And then as our elution buffer is added, you can see an increase in the intensity, indicating that we have collected protein. And all of those red numbers show which tube, so I would know which tube to collect that had protein. So after that, after we have collected our protein, in order to do any kind of experiments, we want the protein to be in the GTP bound, the active state. But currently it's in the inactive state. So what we do is we perform a nucleotide exchange. So we add EDTA in a GTP analog, and we allow it to sit for a few hours so that the GTP can bind. And then we run our protein through a desalting PD10 column. So this desalting column allows any of the concentration of the buffers of the salts to be removed, so that when we measure concentration, it is only due to the protein and nothing else. So once we've run it through the column, we collect it and we perform a Bradford assay. This Bradford assay uses a blue dye and indicates which one of our tubes contain the most protein. So the bluer and brighter the tube, the more protein that we have. You can see to the right a picture of a Bradford assay that we did. And so we collect the tubes that are the brightest, and then we measure their concentration using a nano drop. To ensure that we did all this correctly, we then undergo a gel electrophoresis. So what we do is we use an SDS page gel, and we run our samples of proteins along with the molecular weight ladder. What that molecular weight ladder does is that it has proteins of a known weight that go through the gel, and it allows us to see where our protein falls. So we know at the 25 kilodalton mark, just under the 25 kilodalton mark, is where CDC42 should be at 21 kilodaltons. So if we see bright bands around there, then we know that we have solely collected our protein. So you can see in this gel that there are bright purple bands at around the 21 kilodalton mark, indicated that we indeed have collected pure protein. Once we've collected protein, then it is able to undergo experimentation. And so what we did is we use fluorescence spectroscopy to undergo denaturation studies. And so what we did is that we took our samples of protein and had increasing concentrations of our denaturant, guanidinium hydrochloride, and we wanted to test how the stability of CDC42 and the mutant T35A would be affected by the small molecule. So we did triplicates of these denaturation studies, and this is an example of just one of the protein samples that we did. So you can see that with increasing concentrations of guanidinium hydrochloride, this emission wavelength and intensity of absorbance curve shifts. So at first the peak is around 330, and then as we increase the concentration, it moves to around 350 is where the peak of the curve is. This is indicative of denaturation. So what happens is that tryptophan is in the center of the protein, is not able to emit anything because it is blocked. As we increase denaturation, the protein is able to open up, and so we know that tryptophan emits at 350. So you can see that this curve shows that we did denature, because with increasing concentrations, the tryptophan is being exposed and being able to emit at that 350 wavelength. So we did this triplicates for CDC42 and T35 with and without the small molecule, and these are the results that we got. So you can see with increasing concentrations of our denaturant, guanidinium hydrochloride, we look at the fraction of folded protein. And so our data assumes that at 0 molars of our denaturant, that we have 100% of our protein folded. And then at 5 molars, our protein is completely unfolded at 5 molars of our denaturant. So you can see here that whether CDC42 has a small molecule or not, it still denatures very similarly, indicating that the small molecule may not have an effect on CDC42's stability. You can also see here with T35A on the same graph that the T35A with or without the small molecule, they denature very similarly, indicating that there may not be an effect on the protein's stability with the small molecule. For future work, we were given feedback from our lab group that these samples were not done time dependently, so some samples were subjected to our denaturant a little bit longer than others, and so we want to ensure that all denaturation is done purely based on stability and that time is not a factor. So we would want to do these studies again where we incubate all of the samples for the same amount of time. Somebody else would also need to come in and repeat these experiments using a different denaturant, such as urea, to ensure our data to get another perspective. That would be really beneficial. That is all that I have. I just want to thank a couple people. I just want to thank, first off, you all for listening and for being so flexible during this time, for Dr. Paul Adams and his research group for allowing me to join and for teaching me all these protocols, for my grad student Alix, who's the best and who read every proposal that I ever wrote and has really helped me through my undergraduate career, and then the Honors College for providing me a research grant and for funding my education. Thank you all, and have a good rest of your day.